Hello everyone! The main objective of this tutorial is to learn the names and the functions of the organelles that are found in the eukaryotic cell, or the animal cell. The cells of animals and plants are eukaryotic, as everybody knows, and these cells are usually packed with organelles enclosed by membranes or sometimes by a double layer. And these cells have a nucleus which contains the DNA. So let us see together in details the structure of an animal cell. And this diagram right here shows the main features of an animal cell usually seen under the microscope. So, which structure do you think encloses the cytoplasm? This yellow fluid right here. Yes, it's the plasma membrane. The plasma membrane is the structure that encloses the cytoplasm. It is usually a barrier. It separates the cytoplasm from the surroundings of the cell. It also regulates the flow of materials or substances such as proteins or ions that pass in and out of the cell. Now moving on to the next question. Which structure do you think is the largest organelle? Of course, the nucleus is the largest organelle. The nucleus usually contains chromatin. This is chromatin, as you can see right here. It is a loosely coiled form of chromosomes. And we all know that chromosomes contain the DNA. So, the DNA controls cell activity and inheritance, and it manufactures ribosomes as well. The nuclear envelope right here has nuclear pores, which allow some substances to enter and leave the cells. Which structure do you think is the smallest? Yes, the ribosomes are the smallest structures. The ribosomes are usually made of RNA, and they can be found at the sites of protein synthesis, as well as in the cytoplasm, or attached to the endoplasmic reticulum. We'll see later what's the endoplasmic reticulum. So as you can see here, the ribosomes are responsible for the synthesis of amino acids from mRNA. Okay, now the next question. Which structure do you think appears to be made of tubes? Yes, it's the endoplasmic reticulum. We have two types of endoplasmic reticulum. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Well, the rough endoplasmic reticulum contains ribosomes. They transport proteins and produce vesicles that fuse with the Golgi body. We will see later what's a Golgi body. So proteins are synthesized in the ribosomes, then the endoplasmic reticulum will transport them. However, the smooth endoplasmic reticulum have no attached ribosomes, and the main function is to produce and transport lipids. So as you can notice here, the proteins are synthesized in the ribosomes, then they will enter the endoplasmic reticulum to be transported to the Golgi body. Okay, so now which structure do you think appears to be made of tubes? Yes, it's the microtubules. The microtubules are involved in directing the movement of materials and vesicles around the cell, as well as they play key roles in structural support and cell movement. Moreover, we have the mitochondria. The mitochondria is a compartment within the cell responsible for aerobic respiration. So, mitochondria is usually bounded by a double membrane enclosing a matrix, which have an inner membrane folded into finger-like structures called the cristae. These cristae increases the surface area for aerobic respiration. We all know that aerobic respiration is a process by which cells convert food into energy. So the mitochondria would take the glucose, which is considered here the food of the cell, it will convert it into energy, and the cristae will increase the surface area for aerobic respiration to take place. Now, the Golgi body is usually a structure that collects, modifies, packages proteins from the endoplasmic reticulum. The final two structures are the centrioles and the lysosomes. So usually lysosomes are specialized vesicles that contain hydrolytic enzymes and that can digest old or damaged cell organelles, bacteria, and the whole cell. Centrioles, on the other hand, are made from microtubules and they are used in cell division. And they help in forming the spindle fibers for the sister chromatids to separate. So, finally, these are the main components of an animal cell.